clearing because of viruses that you grew in the lab. It's called a plaque. So when you have, okay, let's say you put a bacteria, a bad strain of strep onto blood, and there's a clearing, that's beta hemolysis. If it's green or partial, that's gamma. If there's no clearing, if it's partial, it's alpha. If there's no clearing, it's gamma. If you are looking at the clearings when you're dealing with viruses, it's called a plaque. Multiplication of the viruses must begin with attachment. Well, if the virus doesn't attach to the host, how are you going to multiply the virus? Okay, the manner in which a disease develops is pathogenesis, because genesis means develop. Seeing how it's transmitted is epidemiology. Getting the disease in a short space of time is epidemic. Endemic is a constant infection. Epidemic is a short-term infection that got spread in a short space of time. And if it's worldwide, it is a pandemic. Okay, so we're looking at 50% of your test population. You want to see how many people are infected. It is ID 50. Viral infections in the blood are viremia. If I said just infections in the blood, it's all of them. But viremia would be viruses, toxemias, toxins. Bacteremia would be a bacteria, and septicemia would be a poison. Typo-parenteral. So the parenteral route, skin route, and mucous membranes, you can get, yes, pathogens. The surface molecule on the pathogen that binds to the receptor is ligand. The cell wall components that can destroy T cells and prevent phagocytosis would be all of them. M, OPA, and mycolic all together can do these. Which component there will bind free iron? Siderophores. Ergotism comes from what organism? Fungi. Toxoplasma, G. lamlia, and trypanosoma, when metabolically active, are trophozoites. And then the scientific study of disease itself is pathology. All right, and let's go to test one. That'll be it. Alright, so which of these is not a shape of bacterial cells? Pseudopods, that's a mode of locomotion. Which of these is the proper way to write your bacteria? C. Italicized genus and species, only the genus is capitalized. Archaea can live in extreme environments. So right there, A and B, you should know, is a thermophile and a halophile, and methanogen, archaea are methanogens also, and they make gas. What's your most typical fungi? Mushroom. A mold. Protozoans use which of these? Flagella? Yes or no? Yes. Fimbria, yes or no? Yes. No. Fimbria is not used. Fimbria are found on bacteria and they're used for attachment. Do protozoans use pseudopods? Yes. Yeah. And amoeba histolica is a protozoan and it does use pseudopod. And the pili, that's for bacterial conjugation. Cell walls of fungi have chitin. Peptidoglycans for bacteria. Cellulose would be for plants. Mycelia and hyphae, those are fungi. And they're not even in the cell wall. So, who coined animal kills and drew them? Lou and Hawk. What Hook did, Hook took slices of cork, and in the slices of cork, because made of dry plant matter, saw little plant cells, and coined the word cellulite. Now, bacteria have what kind of charge? Negative. What's 5,000 divided by 1,000? 5. That's the answer. Distinguishing fine detail would be done via
Resolving power.
resolving power or resolution, tomato, tomato. Okay, this is how I remember number 11. When you're using oil, where you put the oil?
On the side.
On the slide. So the oil in the slide then has to have the same refractive index. That's how I remember it.
So A.
Yeah. 200 centimeters, okay, divide that by 100, you get? There you go. Hook didn't receive credit for the compound microscope. It was Jansen. Lister and Jackson improved the microscope. Jenner came up with the smallpox vaccine, and Van Leeuwenhoek did the animal kills and teeth scraping. What type of microscopy would be less likely to generate radicals? I think so, yes. Two photons. What's a decolorizing agent in a gram stain? Alcohol. Negative staining requires what kind of stain? Acidic. Basic and positively charged are the same thing. Acid fast is for something completely different, for mycolic acid. Okay. Pink oxide, purple bacilli, you have two different species. If you had an old culture, you'd have the same shape but different colors. If the culture was too young, then you would not be able to differentiate gram-negative and positive. And if you forgot a step, well, you kind of sort of can't, because you've got both colors. And it's not an endospore, because endospores are green. Prokaryotic flagellum, which word does not belong?
Microtubule?
Yes, microtubule, and that would be used for the cytoskeleton. Bacteria do not have chitin. Bacteria prokaryotes, I should say, do have a single circular chromosome, do not have membrane enclosed organelles. They have ribosomes, but a ribosome doesn't have a membrane. And the DNA is not enclosed. Now, flagella are not bitrichous. I made that word up. Amphitrichous means flagella on both sides. Peritrichous means flagella all over the bacterium. Lopotrichous means a tuft of flagella. And polar means at one end or the other. What globular protein makes up the prokaryotic flagellum? No. Flagellum. There's no globulin. I made that up.
It was good.
I know. The thing is, like, hemoglobin is also globular. Antibodies are also globular. So globular just means that they look like a little ball. But in this case, it's flagellum. Flagellin are in prokaryotic flagella. So, which of those components are strictly in gram-positive? Walter-Cowic acid. And so this is why when you're doing your problem, you're like, oh, the lipopolysaccharides cause the gram-negative to be beryllium. Well, gram-positive also has lipopolysaccharides. So it's really the Walter-Cowic acid, the gram-positive, that helps it. Okay, so you're doing an endospore stain. The green structures are? endosmolar. The lipopolysaccharides in the gram-negative provide stability, function as an engine, release endotoxins, and also say, hey, I'm gram-negative. So, what's the type of passive process here?
All of them?
Well, diffusion is diffusion, whether simple or facilitated, they will still not expend energy. And osmosis would be the diffusion of water. Okay, tonicity, which one causes the cell to burst, swell and burst? Hypo. Hypotonic means that the solution does not have salt. So the water does not want to be in solution. The water wants to be where it's more salty, relatively speaking. So the water goes into the cell, swells it, and bursts. Facilitated diffusion does use integral proteins. You do not have to expend energy. You can break down large molecules and then bring them in. Not only eukaryotes do it. Indeed. Okay, so if hypotonic swells your cell, which tonicity creates your cell? Hyper. Okay, so glycogen and starch are? Polysaccharide. Starch is sugar. Starch is just glucose linked to glucose, linked to glucose, linked to glucose. And glycogen is glyco, meaning it has sugar. So they're going to be found in the polysaccharide conclusion. If you're aquatic, means you're buoyant,